Good day, everyone. My name is Horacio Vargas Guzman, and this is my fifth SciPy conference, the first virtual one. And today I'm going to introduce to you all Pi for all. Okay, so what this talk is about, it's about Python modeling with Python tools, the structures of macromolecular assemblies, the dynamics of those assemblies, and also their properties. So I'm going to give a brief introduction of multiscale modeling for modeling viruses specifically. And then I'm going to briefly comment about the available tools, Python tools, and what's coming next on that, and make a rationale why we should use Jupyter Notebooks in this field for a collaborative, even more collaborative environment. So what we have currently is structures of these macromolecular assemblies like viruses. And we have also, thanks to experiments, for example, with atomic force microscope, we can see into the dynamics when a certain force is exerted on the apex of such a capsid structure. In this case, we see, for example, that the capsid is formed by different layers. It's a multi-layer capsid. So from the computational side, a couple of decades ago, Klaus Schulten coined this term of the computational microscope, and he indeed simulated a whole viral capsid with all atom resolution, so the highest resolution available in molecular dynamics. And he did it, of course, by using huge supercomputers. So what we have in terms of time scales and length scales, it's, of course, viruses are between tens and hundreds of nanometers, although they are constituted by smaller particles like atoms and a conglomeration of molecules as well. While if we want to talk about time scales, in order to see different phenomena in these huge biomolecular structures, we go from femtoseconds to seconds, even to minutes and more. It depends on which kind of phenomena we would like to see. For example, protein diffusion, it's already in hundreds of milliseconds or seconds. Or we can, for example, by RNA, see the ribose switch functionality. It's also already reaching several hundred of milliseconds. And of course, this means also different modeling scales. It could be atomistic, or it could be coarse-grained, and even these supra-coarse-grained schemes. However, you are asking yourself why I am here at the SciPy conference talking about the natural time scales and the length scales of those biomolecular structures. Well, to model those structures, we need, of course, a very efficient code. And that means mostly a combination of very speedy code like C, C++, or Fortran, and on top of it, a code that allows a very fast development as well, which is undoubtedly Python and the SciPy stack. So currently, we see the left side of the slide. We can, of course, see what's going, where the molecular simulation packages are moving now. They have already, many of them, some interfaces in Python or an API in Python. However, their examples, for example, are not yet completely ported to a Jupyter notebook. And this is where the current status is moving, because everyone wants to give like a notebook format, an interactive notebook shell, like in Jupyter Notebooks, for the whole protocol of simulations, to know exactly what has been done. However, there is still no visible tool that allow us to move on from different scales in the molecular simulation. That means, for example, from the atomistic, as you can see here, this is a water molecule that can be represented with less degrees of freedom by grouping them in only one bit. And then this is exactly the transition from atomistic resolution to a coarse-grained resolution. And this is what has not yet been done, or is not easily doable by just typing some commands on the Jupyter Notebook. And this is exactly the motif of the talk. So we would like to build on top of this amazing stack we have already, this open digital infrastructure, a multiscale, so bridging the scales package, which is Pi for all, which will allow to perform and interoperate between different resolutions in the molecular simulation world. So the modeling premises are, of course, we don't want to stay in a proof of concept basis. We want to go beyond the proof of concept. We would like to model the whole virus with all its detail, and also extrapolate or extrude some other details to a lower resolution, so that we are available to model them as a whole. We would like also to provide and interpret within this tool quantitative observables, such as the free energies, which are basically to see, for example, which structure is more favorable in terms of the whole free energy landscape of these biomolecular ensembles. And on the other hand, we want to go along with experiments, because experimental developments are moving fast, and we don't just get an image of how a virus looks like. Now we can also study experimentally the dynamics of those kind of structures. And of course, try to do it in an efficient way. That means have different resolutions for different regions that we are interested in. That means sometimes we will call the atomistic structure of the biomolecular ensemble, or sometimes we will use a more coarse-grained representation of it. And in this particular case, what we look at is the triatoma virus. And this is a molecular dynamic simulation. This is how the trajectory looks like. In this case, everything is coarse-grained. So we have already bits instead of atoms. And these atoms, as you can see, are resembling the viral capsid of the triatoma virus, depicted with different colors. It's every penton of it. And inside, there are some negative charges in blue, which will produce a repulsive force, and hence it will trigger a disassembly through modeling of such capsid. We saw this great job in the article cited, and then we said, okay, so what about getting the triatoma virus capsid in an atomistic configuration, coarse-graining them with less degrees of freedom, and then starting as it was done, the initial disassembly of such capsid by introducing negative charges, then backmap the structure, and from those structures, since the coarse-grained model is not focused on providing free energy landscapes in this case, we can use another modeling tool, in this case a Poisson-Boltzmann solver, and provide those energies and see if even the structures used for the coarse grain modeling could be possible or are possible energetically speaking. So we do this whole cycle, and as you see, there is first a conversion from atomistic to coarse grain, then is a backmapping procedure, so from coarse grain structures from the initial disassembly paths to atomistic again, and then from atomistic we have to perform other conversions to specifically see the charges of each of the atoms and also the van der Waals radius, and then we can use a continuum solver for that, which is in this case Poisson-Boltzmann. So for the modeling, what we did, I'm not going to go in any detail, but basically with the Poisson-Boltzmann method, we solve the polar part of the electrostatic energies, and we have solvation energy given by this reaction field, the integration of reaction field, and a Coulomb energy given by Coulomb's equation. And then we tackle specifically 3 cases with different boundary conditions. So first we have a region inside the capsid, region 1, a region outside the capsid, region 2, and then we have a region inside the capsid. And in the case of scenario A, we will see that we have the structure not disassemble the compact structure of the capsid. Then we have in scenario B a compact structure as well, but already with negative charges inside the capsid. And then in scenario C we have the initial disassembled structure of the capsid with negative charges inside as well. So what we observe easily by performing these Poisson-Boltzmann calculations is that the structure of the initial disassembly, it's possible energetically speaking, because it gives us even lower free energy than the structure, the compact structure of the capsid with negative ions inside. So it means the capsid relaxes, it's in a more favorable energetically conformation when it starts disassembling once this artificial repulsive force is introduced by negative charges. So going back to Pi4All, the Pi4All is like a multiscale molecular glue. It's a within the whole Pythonic ecosystems. So we have as an input, of course, the molecular dynamics trajectories from different packages, LAMMPS, Chromax, Espresso, and so on. We can also recently with the COVID pandemic, the Molecular Software Institution and the BioExcel, for example, in Europe, they agreed to provide all the molecular simulation trajectories to share among scientists all over the globe. And we have, of course, as well the Protein Data Bank, which tackles this, which has already many thousands of different structures of biomolecular ensembles, and many other packages like QMD and so on. And once we have these trajectories, what we would like to build, this is the main goal of Pi4All, it's analyze in a first instance the free energies of those structures, but also interact between the different representations, if they are atomistic, if they are coarse grain, or if they are even continuum representations of those structures. And through this multi-scale interaction, we would, of course, we have the aim to simulate the whole virus with their glycoproteins, with the genetic material, with their capsids, their envelopes, and also perhaps interaction with the human cell enzyme like the AC2. And all of this now currently is done by using PICB, using NanoShaper, using PDB to PQR, and many other tools which are also Python based, and then perform the analysis by also knocking the doors of the Pythonic ecosystem, this open digital infrastructure, using MD analysis, for example, scikit-learn. We can use, of course, pandas and many SciPy tools, and of course, plotting with Matplotlib. So this is the overview of what Pi4All should enable. And the take-home messages, of course, are that the Pi4All will provide free energy calculations in even very small clusters, or a GPU workstation, or even in the cloud if you have access to a GPU. So you don't need huge clusters anymore, because first of all, many of the trajectories are going to be shared through, for example, the initiatives of MolC and BioExcel. And also, we are modeling here multiscale schemes. That means we are abstracting some of the atomistic details of the structures and making more feasible computationally. And this is done systematically and rapidly through Pi4All, as you can see now in an example. So we first, for example, connect to a cloud server with an available GPU. Then we import the NumPy SciPy Matplotlib as a basis. In this case, we are installing some other libraries that are not there. The Swig for using PICB. Then we install PICB, and then we import the Pi4All library, and by just two command lines, so Pi4All compare pHs, we can say, okay, we would like to compare the SARS-CoV-2 with the SARS-CoV-2 without the human enzyme, and then compare also the free energy values with the older SARS-CoV coronavirus from 2002. And this comparison should be done, in this case, for pHs from 5 to 8. And then we do a plot pHs in the x coordinate. We have the structures we name. In this case, our structures coming from MD simulations, all atom simulations, 320 nanoseconds. It doesn't mean that Pi4All is performing the structures, but it has already a database with the structures inside. And then in the y-axis, we plot the pH, and in the z-axis, the free energies. And then we can easily compare already if for example, SARS-CoV-2 has even less free energies. That means the structure is even more in a lower free energy minima, and so on. And then we can perform different analysis. So the idea will be, of course, to simulate even more than the glycoprotein, as it's the case from this example, as we are calling here the structures of the glycoproteins of both SARS-CoV, to compare and see which, for example, has a higher affinity, or why it's SARS-CoV-2 more widely spread in comparison to SARS-CoV, which was also producing contradictory results in sense of experiments, because some experiments said that the the new coronavirus SARS-CoV-2 has a higher affinity than the older one, and others say that those differences are not big. But we can see here from a more theoretical overview. And of course, in the next step, we would like to enhance this structure to the whole envelope of the virus, and why not even put it inside the genetic material. So I would like to acknowledge all the funding agencies to perform this work, the Slovenian Research Agency, the TRR 146, and the institutions where I have been working on these ideas, and of course my collaborators all over the globe, and all of you for your attention. If you have any questions, please contact us, or me, or Christopher Cooper as well, or just leave a message, a note, or clone our GitHub page. Thank you very much.